I'm at Randolph-Macon College in biology. Randolph-Macon is about 1,500 students, and we're just north of Richmond, Virginia. And I teach all levels of molecular biology and genetics, all the way from intro to upper levels. So I think we're all here today because we all know that using the primary literature in classes is a challenge for many reasons, many of which we've talked about already today. Just the vocabulary, both regular, challenging vocabulary, as well as scientific jargon. I know I find myself looking up words when I read scientific papers myself. Rarely is enough detail given in the introduction for undergrads to actually understand the science they're reading about. They may not be familiar with the science and or the techniques. And one other reason it's challenging for students to get out of papers what you want is because the authors have already done the interpretation for students. And so they read that and then they have their blinders on, and that's all they can see. So reading the papers is a challenge. So what I use as a strategy in some of my classes is to eliminate some of these barriers by not asking the students to actually read the paper, but still using the paper. So that takes out the vocabulary issue. I replace the introduction with other readings, and they don't have the author's interpretation because I don't let them read what the authors have written. So that leaves focusing on interpretation of the data and the science and the techniques, which is what I want when I use this particular strategy. So what I'm using is basically a flipped approach to the scientific literature. And if you're not familiar with the flipped approach, that's giving students the easier parts of what you want them to learn outside of class, before class. And then when they come into class, you focus on the harder things and more upper level thinking. So in terms of the scientific literature, how that works is what they read before class are, textbook readings, YouTube videos related to the background of the paper. So that's the easier part they do on their own outside of class. And then in paper is when we discuss, or in class, what we do is discuss the paper using an active learning approach. All right, so this slide is going to outline the 5 steps I've broken this strategy down into at a very superficial level. And then I'll go through these 5 steps again with a specific example of a paper that I've used in a class. So the first step is the only one that occurs outside of class, and that's giving them readings ahead of time, basic readings. The next ones occur within class. So those readings I give them before class usually don't cover everything they need to know from the background of the paper. So I start by filling in the gaps. And since I'm a molecular biologist, these usually relate to molecular details that might be more difficult for students to understand. So that's what we start with, kind of me presenting, which is not very active. But then the next step is where students really become active. This is where students brainstorm. So I usually set up the background in a way that gives students an idea of where we're going with this information. So students at this point start brainstorming either about experiments to be done, if I think they're at a level where they can propose experiments, or maybe they just come up with scientific questions at this point if they don't know how even if they don't know how to address those questions, but to start thinking about what kind of science would be done. Next step is, there we go. Next step is also very active. This is when I actually parse the paper apart, choosing particular figures, and I show those figures and give student groups time to interpret the data. So at this point, they still haven't read the paper. They don't have any of the text. I don't even give them the figure legends. So they are really relying on their own skills to interpret the data. And then the last step, I like to end with some type of synthesis activity, which is usually writing an abstract based on the data that I've shown. All right, so those are the 5 steps. This is all done without actually reading the journal article, but they are still getting information and seeing the scientific process in a recent journal article, even though they're not reading it. Okay, like I said, I wanted to share with you a specific example of how I've used this strategy. So I'm going to talk about a paper that I used in an upper level class based on molecular biology of human health conditions. And what I'm going to talk about today, I've done in a 90-minute session, more or less. Sometimes I run out of time. This is the article that which this particular one is based on. It's a Nature article, I think from 2013, where they've taken the molecular mechanisms that turn off one copy of the X chromosome in XX individuals and use those molecular mechanisms to potentially turn off one of 3 copies of chromosome 21 in a cell culture system. It's a pretty complex, very molecular paper. This is an upper level class, so students do have some knowledge of some topics ahead of time. When I introduce this paper, we've already talked about trisomy 21, Down syndrome. We've already talked about how genes get overexpressed when there's 3 copies of them, but they know nothing about X chromosome inactivation. All right, so these are the 5 steps again. First step, since they don't know anything about X chromosome inactivation, that's their take-home assignment. I give them this Nature citable reading on X chromosome inactivation, which is at the appropriate level. So they're not at home frustrated because they don't understand it. It's written at their level. And it contains some molecular details about X chromosome inactivation, but not at a level they can't handle. And I give them some study questions to go along with it, so they focus their attention on what I'd like them to focus their attention on. So that's step 1 outside of class. Then when they come to class, this is when I fill in some of those gaps. We start by just going over the study questions to make sure everyone understands the basics of X chromosome inactivation. And then I get into a lot more detail, especially about the molecular details involved. So, and this part of the discussion is based on a review article that is way too in-depth for the students, which is why I don't discuss it. But this is a figure from that review that I actually show in class, showing that there is a gene here in the X chromosome inactivation center that expresses non-coding RNAs that bind to the X chromosome and condense the chromatin in that region. So that's what they get ahead of time. So then this is when I put students to work brainstorming. They have the background. They usually work in small groups because that gets their brains flowing. And still at this point, I don't necessarily ask them to come up with particular experiments. And for this one, I actually give them some guided questions to work on. So this first question here is thinking about putting these topics together. So XS gene is the gene that expresses that non-coding RNA, and thinking about how that might come together with trisomy 21. And then I ask them to make some predictions about what they might see. And they have the skill, they have the knowledge to answer these questions, because the answers there are many answers that were in the readings that they did. So they can answer these questions. And here we go. And as they're answering this third question in particular, because this is forming the basis for the data I'm going to show, I actually make a list on the board. So as students are giving me answers, and there are many, 6 to 10 answers that they could give, I write them on the board and I leave them up there for students to see. After this brainstorming and discussion, that's when I start revealing data from the paper. And I do one data set at a time so they can learn from the first data set. I don't provide the figure legends, like I said, I do provide annotation of the figures if necessary. So here's an example of a figure that I've shown in class from this paper. And this one's actually pretty well annotated already, but I provide a little bit of extra information about, that this gene is actually on chromosome 21. And then I don't just set them loose and say interpret it. I, again, give them specific questions. So these are questions they can answer based on this figure. And then we talk about it, and then we'll do this. We'll repeat this process for multiple figures from the paper. And you can see here, even for this figure I didn't even give them the whole figure. This is just panel C because this is from a Nature paper, which is probably too difficult for our students. All right. Importantly, I wanted to remember to say this, as we're going through this data, I'm checking off that list that I created on the board of student ideas for ideas that could be tested. So they're actually seeing that experiments that they thought to do, they have actually been published, and they get to interpret the data and see the results. So that is often very exciting for them to see their ideas are actually valid science ideas. All right, and then assuming I have time at the end, we do a synthesis activity. And like I said, having students write an abstract without the aid of the paper is probably my favorite assessment. It really tells you what the students got out of what they did in class that day. And it's not necessarily an easy assignment, so I don't necessarily expect perfection from them. All right, so I think there are lots of advantages to this approach. One is just that students do a ton of higher level thinking during class that day. They're developing scientific questions, hypotheses, maybe designing experiments if they're at the right level and they have enough background. They definitely analyze data and draw conclusions on their own, and they don't have that crutch of what the author already interpreted for them. I also like looking at scientific papers in this way because there's lots of opportunities for success for them. And thinking about many journal clubs where the only way they're analyzing the paper is by critiquing something that experts have done, that's hard to come up with something that these experts haven't already thought of, right? But with this, they can propose anything, and many are very valid ideas and that were actually done in the paper, so that they get a chance to have that success with science. In that list that I make, there's often experiments proposed by students that weren't in the paper, and some of them are very valid experiments they proposed, so maybe they said, well, why didn't they do that experiment in the mouse? And so that gives us a chance to talk about, how science is actually done and, how fast science moves. This strategy definitely allows gaps in knowledge to be identified. If students don't understand what the results of a figure are, I'll know right away if they can't discuss that in class. So we can definitely see where the gaps are because, like I said, they don't have what's written to guide them. And I would say with this strategy, there's a lot of investment in the paper. Even though they're not reading it, they have fun interpreting the data and making predictions and seeing their success. And one thing I really like about this strategy is that it can be adapted to all levels, all scientific disciplines. You can tailor the questions for both the brainstorming phase and the interpretation phase. You can tailor that to the level of your class. If you have a higher level class, you can ask students to make predictions about control data, talk about what kind of controls could be shown. Or maybe lower level classes, you feed them some of what they need and let them go from there. And you can definitely adapt this to any amount of time by showing more or less data, adding fewer or more writing activities at the end. So it's very adaptable. And I think I will stop there so I don't go too far over time. Thank you all for your attention.
Wonderful talk. We got a few questions already in the chat. First being, do you have some preliminary discussions on the techniques used in the paper, or or how to read the graphs, the gel, or whatever is in the figure?
Yeah, that's a good question. Yes, sometimes in that background part where I'm filling in the gaps, some of what I fill in sometimes is what's actually the techniques that they use. And sometimes I give that as they read about those techniques in their take-home readings ahead of time.
And this one speaks to me. What if they don't complete the homework?
Well, I usually have them, I do kind of random sampling. I grade some of their pre-class assignments. And so they think that it could be graded at any time. Yeah, and I do teach small, I have the advantage of much smaller classes. And so they usually do complete them. And I'm sorry, I can't help more with that problem. I wish I could solve that problem.
So this is a good one. So do students simply go to the paper? Like you distill it down, but do some like go, I'm going to go look at the paper and see what the author said about this?
No, because they are doing it all in class.
Okay.
So they don't really have that opportunity to do that. I mean, you know, cheating can happen at any place online, but no, I just don't let them. And even the writing the abstract, I've found that it doesn't work if you send it home, because then they do look at it. So if you want them to write the abstract at the end, you have to allow that 15 minutes or so. I know that isn't much to write an abstract, but they can put some ideas down. And
I'm also on this, like I like the idea of having students apply predictions. Think a step ahead. Do they ever try to short circuit the process? Oh, and we just answered this question. I just reread the rest of it. So yeah, so they don't have time to, but do they talk amongst themselves, or is it all?
Yeah, and it's fine. I encourage them to talk amongst themselves, because when they're talking to each other, they're learning whether they're getting the information or whether they're talking about the information. So I definitely do steps 3 and 4 as a group. Writing the abstract at the end, I have them write their own, but I'm not opposed to chatter as they work on it, because they're not just copying from each other.